So we're gonna do the chapter 3 biology online quiz or course practice. Okay, so question 1, the diagram below shows how a large molecule is converted into two smaller molecules by the help of molecule P. Which of the following is correct, or statements are correct about molecule P? It is a substrate? No. It consists of nucleic acids? No. Okay. Here in the image, you can see that it is an enzyme. Enzymes are catalysts. They're also actually known as biological catalysts, and they are made of proteins. We'll choose the correct substrate that X would most probably work on. They would have to fit pretty much perfectly, and the only one that would actually fit perfectly in the whole of the enzyme would be this one. So that's it. Why do most chemical reactions occur faster at higher temperatures? The enzymes move faster. The enzyme is denatured. No, sorry. The enzyme moves faster. The reacting molecules bump into each other more frequently. Yes. The reacting molecules move around slower. No. The reacting molecules have more kinetic energy. Yes. The reason why they actually move faster, making them bump into each other more frequently, is because they have more kinetic energy from the heat or the rise in temperature in general. Now, here, okay, step 1, enzymes have a defined structure. They are made of large, complex protein molecules because enzymes are proteins. The site at which an enzyme binds to its substrate is called the binding site. Quite self-explanatory. Enzymes are extremely specific to the substrate they bind to. They bind to these molecules for a short time, forming a complex. Okay. The molecules upon which an enzyme acts are converted into different molecules called products, because that's how the reaction works. So the substrate reacts with the enzyme, causing the enzyme to break down the substrate, which gives us products. So why are enzymes important for living organisms? They participate in chemical reactions and are transformed into new products. No, in fact, enzymes do not transform into new products. They are not changed. Only substrates are. They bind to several different substrates at the same time. No, they only bind to one at a time and to very specific ones. They ensure that metabolic reactions occur quickly enough to sustain life. Yes, it is their job and their purpose. They are broken down, which is why they're known as biological catalysts. They are broken down into sugar molecules that serve as a source of energy. No. They provide heat and insulate organisms from the external environment. No. Okay, choose the correct answer to identify the factor affecting enzyme activity described in each of the following cases. When it increases, it speeds up the movement of the enzyme and substrate molecules, thereby increasing the chances of forming an enzyme-substrate complex. This would be temperature. The more it is present, the more often it will react with enzyme molecules, and the more products will form. This is substrate concentration. It determines the level of alkalinity or acidity. This is pH. Okay, so to explain it, or to have a short summary, the higher the temperature, the quicker the molecules will move, which will lead to more collisions, or they will bump into each other more often, which will increase the reaction rate. Now, Another thing that affects the activity of enzymes is concentration, which basically the higher the concentration, the more molecules that you have of this. So let's say the higher the substrate concentration, the higher amount of substrates you have, which will increase the reaction rate because it will increase the possibility of a substrate and an enzyme coming together and going through a metabolic reaction. And finally, the level of pH, or how acidic or basic something is. And another word for basic is alkaline, I think, or here they said alkalinity. So the level of pH can affect the activity of enzymes depending on the enzyme. And that's it basically. So yeah, that's basically just a summary of everything that can affect the activity of an enzyme. Which enzyme carries out each of the following functions? Answer with one word. Breakdown of proteins. Protease. Here, okay, wait. Let me just breakdown of lipase. Breakdown of sucrase. And basically, as you can see, there's a pattern for almost every single thing that enzymes break down. They are named based off of what they break down. So like here, the enzyme, which is called protease, breaks down proteins. So you just take the name of whatever it's breaking down, remove the last three letters, and replace it with ASE, or A's. Now, of course, there are a couple of what are they called? Exceptions, but the main exception is starch, and amylase is what breaks down starch. The following diagram represents an enzyme catalyzed reaction. What theory of enzyme substrate reaction is represented in the diagram? Here you can see that the enzyme is not perfectly shaped for the substrate, but the enzyme changes its shape so that the substrate can actually fit into the enzyme, and so that the reaction can take place. So this is called the induced fit theory. What does each label represent? Here, label A represents the substrate, label B represents the enzyme, and label C represents the product. The substrate being what the enzyme reacts with, the enzyme is the enzyme, and the product is what the substrate turns into or changes into or produces at the end of the chemical reaction. Okay, the following graph represents the action of two enzymes, gastric protease and intestinal protease. What is the optimum pH for gastric protease? Okay, here you just have to read the graph. Basically, it's not too hard. The dotted line is gastric, so you just have to check where is the top of it. So I'm gonna try to write this somehow around here, right? So the top of the hill, all the way to the top, the very top of the hill. I'm horrible at the drawing part. I'm just gonna give up on that. But the top of the hill-like structure or line is the optimum pH, which is the pH at which the enzyme works the best at. And for gastric protease, which the dotted line is 3. For intestinal, right? Yes, intestinal, is the normal line, which is 8. And here you can see the enzyme that works better in the stomach, where the pH is blank, is the blank. Okay. Here you have to see which one works better. And generally the gastric protease works better, and you can tell this by looking at the graph, because the gastric protease is closer to the line, is shorter, and here this line shows the rate, and around here, like any other graph, it would be like 1, 2, 3, and so on and so on. So, but I would say now I might be wrong, but how I found out that it's gastric protease and not intestinal protease is because it's shorter, showing that it's smaller and generally takes less time than intestinal, which shows that it works better. So where the pH is acidic, because here gastric protease is 3, and 6 till 0 or 0 till 6 is acidic, 7 is neutral, and 8 till 14 is basic on the pH scale. And so gastric protease. So yeah. Next question. The following diagram represents an enzyme catalyzed reaction that occurs in the human body. What would substrate A most probably be? Okay, considering the fact that it produces amino acids, substrate A would be protein, because protein breaks down into amino acids. What would enzyme B most probably be? Protease, because, like I said before, protease breaks down protein into amino acids. Lipase breaks down lipids into glycerol and fatty acids, and so on. There are many examples, but yeah. What would happen if B was subjected to a very high temperature? A very high temperature would mean that it was like around above 40 degrees Celsius. And if it's above 40 degrees Celsius, then the enzyme will become denatured. And when it becomes denatured, it loses its bonds or cross linkages, which will cause it to lose its shape, which will cause it to lose its function, meaning that it won't work anymore and it won't be able to work again. It will be permanently denatured. So, if it's a very high temperature, the protein would be digested slower or not at all. So yeah. Next question. Starch can be broken down in humans and other mammals. Enzyme A breaks down starch at a neutral pH in the human body. A is most probably... Okay, well, we know it breaks down starch. And starch is broken down by amylase. So here in the examples, there's salivary amylase and pancreatic amylase. And how to know is that they said at a neutral pH. So neutral pH means a pH 7. And this you have to memorize it. Salivary amylase has an optimum pH of pH 7. Or a neutral pH. So yeah, here, right? And this is just generally something you need to like memorize. Salivary amylase works best at a pH of 7 and a temperature of 37 degrees Celsius. And enzyme A catalyzes the reaction of breaking down starch into maltose. Starch breaks down into maltose. That's just something you need to memorize, something you need to know. Just like how proteins give us amino acids, lipids give us glycerol and fatty acids, and so on. Okay. Drag and drop the correct description for each of the following graphs representing the effect of different factors on each activity. Okay, so on enzyme activity. So for this one, you just have to read or see the graph, see what it gives you, what it tells you, and then just drag and drop the correct answers. So in A, you can see that the highest point of the thing is 50 degrees, because here you see temperature. So you say here, an enzyme with an optimum temperature of 50 degrees Celsius. Optimum means it works best at. So optimum temperature means that it works best at this temperature. In this example, it would be 50 degrees Celsius. Although generally at 50 degrees Celsius, enzymes are usually getting or becoming denatured. And of course, optimum also is used for pH. So the optimum pH would be the best possible pH for that enzyme. Next, here, like I said right now, the best possible pH is the optimum pH. So here in this one, the optimum pH is 9, an enzyme that would be most effective at pH 9. Here, you just have to read the graph and pick the answer. Here, you can see that the pH is at 3. It's the highest at 3. So you go here, and here they didn't say pH 3, but they did say an enzyme that works in an acidic environment. And this generally works in an acidic environment because the environment that it works in is less than 7. So generally it works in an acidic environment. So yeah. Next question. Put the following steps of formation of sucrose during an enzyme catalyzed reaction in the correct order using numbers. So the first step of enzyme catalyzed reactions would be first the substrate, which in this example would be the one glucose molecule and one fructose molecule, binding to the enzyme or the active site of the enzyme in general. So that's 1. 2 is an enzyme substrate complex is formed for a short while, because once they bind together, that is the complex or the molecule that is made, enzyme substrate complex. Then afterwards, sucrose and water are formed. In other words, the products are formed and the enzyme is freed and takes part in another reaction. Next question. Match each of the following enzymes to their corresponding use in biotechnology. Okay, so biotechnology is just using biological things in industrial things. So, or more specifically, when biological processes are used for industrial ones. Here, they're asking to put or match each of the following enzymes to their corresponding uses in biotechnology. Here, let's see. So, breaks down cellulose into sugars that can be fermented to produce fuel, cellulase. Cellulase breaks down cellulose, like how protease breaks down proteins, and lipase breaks down lipids or oil, because oil is lipids, is a type of lipid. And here another protein, protease. And generally, actually, this is a very good thing to memorize, to just memorize what cellulose is used for in biotechnology, what protease is used for, what lipase is used for. So yeah, definitely recommend just memorizing that on your own time if you can for the exam. So yeah, what is the name of the enzyme? Oh, and sorry, I forgot to explain it. Protease is used in biodetergents to break down proteins or stains caused by proteins, like I said here, and into amino acids, soluble amino acids, so that water can wash them away. And it's the same thing for lipids, except lipase breaks lipids into glycerol and fatty acids, which just like amino acids are washed away when the clothes is washed. after using the biodetergents. And cellulase breaks down cellulose into sugars that can be fermented to produce fuel. So, yeah. Now, here, this is in practical work 3.4. There are also other, what are they called? Other experiments that are mentioned in 3.4. Definitely recommend learning them because I don't think any other ones come in the online quiz, but it is good to know them in general. But anyway, here, okay, refer to the reaction below to answer the following questions. What is the name of the enzyme that catalyzes this reaction? Okay. The name of the enzyme that catalyzes hydrogen peroxide is called catalase. Catalase breaks down hydrogen peroxide into water and oxygen gas. How is the presence of the oxygen produced tested? It is tested by lighting a wooden splint, then blowing it out, letting it cool, then putting it at the mouth of the test tube to see whether it relights. If it does, then it will prove that oxygen is produced by the catalase breaking down the hydrogen peroxide. But here, the wooden splint that was lit up before will now be called a glowing splint, because once you light it up, it starts glowing. And it will glow. Okay. Human saliva contains an enzyme known to break down starch into maltose. This enzyme works best at 37 degrees Celsius. What is this enzyme called? Okay, so it breaks down starch, so it's called amylase. You just gotta memorize it. If it breaks down starch, it's amylase. If it breaks down proteins, it's protease. And if you ever forget, just take the name of what it's breaking down, replace the last 3 letters with A's, and you're good to go. Except for starch. Starch you have to memorize, it's amylase. Now for this one, the molecule on which the enzyme acts in a chemical reaction is called a substrate. Enzymes are extremely specific with respect to the substrate they bind to and to the chemical reaction they catalyze. This means that an enzyme that normally acts on one substrate will not act on a different one. They speed up chemical reactions taking place inside living cells without getting consumed in the reaction. This is actually a very good summary of how enzymes work. Of course, there's more to know. Either way, using the graph below, show how the human salivary amylase enzyme behaves when increasing the temperature from about 10 degrees Celsius to 60 degrees Celsius. Okay, which letter represents the human salivary amylase enzyme becoming denatured, and which letter represents the human salivary amylase enzyme completely denatured? Okay, so here, you just have to read the graph basically. Point A, it starts going downhill, meaning that it is becoming denatured. As when it's completely denatured, it means that it has gone completely downhill and it's all the way down at the bottom and it just isn't working anymore, which is point C. The graph below traces the relative rate of enzyme action of 2 enzymes, gastric protease and intestinal protease, at different pH levels. Okay. What can be observed regarding the relationship between pH and the activity of each enzyme? Drag and drop the correct answers to complete the paragraph below. Okay, so the pH is a factor that affects the activity of enzymes. Most enzymes work best at certain pH intervals. Gastric protease works best in blank conditions, reaching its optimum activity at around pH blank. Here you have to check. Gastric protease is the dotted line, so it works best at around 3 you can just read the graph. So here at around 3 pH, and like I said before, 0 to 6 acidic, 7 neutral, 8 to 14 basic or alkaline. It means it's the same thing, just different way of saying basic. Now, intestinal protease works best in more blank conditions, reaching its optimum activity at around pH blank. Here you can see intestinal protease is the red line, so 8, at around 8, pH of 8, which means basic conditions. What would be the consequence of mixing both enzymes and their substrates in one test tube with a pH between 3 and 7? Choose the correct answer. At pH blank, both enzymes would have some limited activity. So they said with a pH between 3 and 7, so 5. It's not very complicated, it's just between 3 and 7. And yeah. The graph below shows how the human salivary amylase enzyme behaves when the concentration of the substrate changes. Okay. Drag and drop the correct answers to complete the sentence below. When the concentration of the substrate increases, the rate of the reaction increases until reaching a limiting rate, which further change in the substance concentration produces no significant change in the reaction rate. Generally, hiring or increasing the concentration of substrates or enzymes increases the reaction rate, but to a specific point. Like if you increase the concentration of substrates, it will keep the reaction rate will increase until a specific point. And it's the same thing with enzymes, each one having their own limits, and it will come in another question, so I'll save you the explanation twice and you can just wait until I think around question 25 or 26, because I know I'm gonna explain it then anyway, and I don't want to explain it twice because I feel like that's kind of a waste of time and kind of annoying. So okay, so the picture below illustrates an experiment used to test the breakdown of starch by the human salivary amylase. What is the product of this reaction? So it's breaking down starch, so you get maltose. Here, underline the tubes that would contain the product of this reaction after 20 minutes. After 20 minutes. So 20 minutes is a relatively long time, but although this might actually be a little bit confusing, it's even a little bit confusing for me. The tube that would go through the reaction the fastest would be tube 4, because it has amylase and it is being heated to amylase's optimum temperature, which is 37 degrees Celsius. So yeah, tube 4 would contain the product after 20 minutes, because 20 minutes is a long time, and it is probably going to be one of the only ones that have actually produced the product or finished the reaction in the span of 20 minutes, and the other ones would either take longer or in general take longer, or in general they will take longer than tube 4, but generally they might take longer than 20 minutes. That's what I personally understood, and I just hope that made sense. The table below shows the result of an experiment done to determine the effect of pH on enzyme activity. Okay, which tube has the most alkaline solution? Okay, alkaline means basic, so from 8 till 14. So which one has the highest pH, basically? Just check the pH. 3, 6.8, 7, 7.5, and 9. 9 is the highest, and the one with pH 9 is test tube 5. So just put test tube 5. At which temperature should all tubes be placed? Okay, let's see. So 0 degrees would take forever. It wouldn't do it any good. And if anything, it would just make the reaction rate slower. 100 degrees Celsius would just denature the enzyme, causing it to lose its function and never work again. And 37 degrees Celsius is generally a really good, temperature for most enzymes, but specifically for amylase, it's the best temperature to have it at, which makes it the optimum temperature of amylase. What is the molecule present in the test tube after the action of amylase? After the action means the product. So what is the product? Basically, that's what they're asking. Maltose, because amylase breaks down starch and starch produces maltose. What is the optimum pH for this enzyme? Optimum pH. So which pH worked the best? So we check which one took the least amount of time. The one that took the least amount of time was test tube 2, and it had a pH of 6.8. So that's the optimum pH for this enzyme. So basically, you just have to read the data given and get the answers from there. Experiment below represents the action of an enzyme on hydrogen peroxide. I think it was question 18 or something. It was like a question from a long time ago. But the point is, like a question before, this is an experiment that is in 3.4. Definitely recommend looking over them again. They're very important, and they will definitely come in the actual exam. But anyway, name the enzyme present in the liver that breaks down hydrogen peroxide. Catalase. Catalase is what breaks down hydrogen peroxide. Other than water, name the product of the breakdown of hydrogen peroxide. The product would be water and oxygen, so O2. Okay. In which experiment, A or B, is the enzyme denatured? In B, because B gets boiled, which causes it to become denatured because it's raised to a very high temperature. A glowing splint is put near the mouth of the test tube in both experiments. In which experiment, A or B, will the glowing splint relight? In A, because B was boiled, which caused the enzymes to become denatured, which caused the reaction to never actually take place, which meant that there was no products, and the products is what relights the glowing splint, or to be more specific, oxygen. Next question. Some stains on clothes, like egg yolk stains, are not easily washed off with ordinary detergents. They contain insoluble colored fats which get stuck onto the fibers of the cloth and will not wash out. To help get rid of these stains, biological powders are used. Okay, so using the information above, fill in the blanks to explain how these biological powders work. Biological powders contain the enzyme protease, which breaks down protein specifically for fats. Sorry, I thought this was proteins. No, they're asking for specifically fats. So lipase breaks down lipids into soluble fatty acids and glucose. Was it glucose or glycerol? No, glycerol. Sorry. Glycerol. My bad. Sorry. The shape of the active site will only allow one type of substrate to fit. The enzyme is said to be specific for the substrate for an enzyme mediated reaction to occur, the blank and blank of the substrate must be compatible with the blank of the enzyme. Okay, so the shape and charge of the substrates must be compatible with the active site of the enzyme. Now this could, I think it's supposed to be compatible and not specific, but it could be, it has the same meaning honestly, which is really annoying because I might get it wrong because I used compatible instead of specific, but anyway, not the point. Anyway, the rate of an enzymatically controlled reaction blank as substrate in concentration increases, I would say increases, as the substrate, as like I said before, this question and the one right after it do give a really good summary of the whole concentration thing. As there are more collisions, sorry about the background noise if you can hear it, collisions between the substrate molecules, between the substrate molecules and the enzymes. Therefore, more product is formed per unit time. When all the something are fully occupied with something, increasing, when all the I think enzymes are fully occupied with substrates, although this could be active sites instead of enzymes. Again, same meaning, it's just, the online quiz has very specific answers. But yeah. And the rate of an enzymatically controlled reaction blank as the enzyme concentration increases. So increases as the enzyme concentration increases, as there are more collisions, just like the one before, because the more the molecules, the more likely they are to combine, because the more they bump into each other and everything. So yeah. Sorry for that. Okay, let me say that again. The higher the concentration, the more molecules there are. The more molecules there are, the faster the reaction will take place, because the more likely the enzyme will meet or attach itself or bind to a substrate. Yeah. Now, when all the blank have combined with the blank, I would say substrates have combined with the active sites, of the enzymes, increasing the concentration of the enzyme will no longer increase the rate of the reaction. Enzymes are proteins. This is just a fact. The precise three-dimensional shape of the enzyme is determined by the amino acid sequence, just like proteins, because they are proteins. The shape of the active site of the enzyme must be complementary to the substrate on which it is to act. Slight changes in pF affect the activity of the enzymes due to the alterations in the active site. But these effects are usually irreversible. Extreme changes in pH will denature enzymes, which leads to loss of its functions. This is irreversible because when the enzymes become denatured, it just is not, you can't bring them back to how they were before. And it's the same for proteins, which is because enzymes are proteins. Most enzymes work best at a particular pH known as the enzyme's optimum pH. Optimum pH. The enzyme pepsin found in stomach works best at pH of, I think it is 2. Now the book doesn't specifically mention the pH, the optimum pH, or at least I don't think it does, but generally I think it is 2. Okay, so I checked the book really quickly. I paused the video and checked the book, and yes, it is in the book. My bad, sorry. It is in the book. The pH, the optimum pH is 2 for pepsin, and for amylase it is 7. This is obviously something that you just have to memorize. And which of the following can be used to measure pH? pH meter, universal indicator pH paper, and litmus paper. And yeah, just checking if that was it. That is it. Honestly, basically it's pretty self-explanatory. Honestly, pH, and litmus paper, and that's it. Identify whether each of the following is descriptive of acidic solutions or basic solutions. Has a high concentration of hydrogen ions is acidic, because the more hydrogen ions, the more acidic. Has a pH greater than 7, basic, less than 7, acidic. And of course, as a little reminder, if it has a pH of exactly 7, then it is neutral. And turns red litmus paper blue is basic. Turns blue litmus paper red is acidic. This is something that you need to memorize. So when it turns red, it's acidic. When it turns blue, it's basic. pH is used to express the blank of a solution. pH is used to express the blank of a solution. pH is an indicator of the concentration of blank ions in a solution. Okay, here is acidity. So it is used to express the acidity of a solution. Here it is an indicator of hydrogen ions, I believe. And from 0 to 14, you have to memorize that. 0 to 14. It's actually, we did it I think like last year or something. Either way, the enzyme blank breaks down blank into sugars that can be fermented to produce biofuel. This was in 3.3, I think. And basically, you just have to memorize like the biotechnology. So you have to memorize like which each one is used for. So in biotechnology, we're using biological processes to do industrial ones. And for this specific example, the enzyme cellulase breaks down. Cellulase breaks down. Cellulose into sugars that can be fermented to produce biofuel. And here's another example of like biotechnology. Biodetergents contain enzymes to remove stains from fabrics. Blank help remove stains caused by proteins. Protease help remove stains caused by proteins present in substances like milk, blood, and egg by breaking down insoluble proteins into soluble amino acids, which then dissolve in the washing water. Blank remove oils stains by breaking down fats into blank and blank. So here, lipase removes oil stains by breaking down fats into fatty acids, and glycerol. Okay. It is best to use these biological washing powders at an optimum temperature of 40. So this, you just have to memorize it. I don't actually think it was mentioned in the book, but you do have to memorize it. Optimum temperature of 40 degrees Celsius. It might come in the exam, it might not, but yeah, it's just good to know it anyway. So yeah, the enzyme blank can catalyze the hydrolysis of blank found in fruits, allowing the easy extraction of juice from fruits. So this one is pectinase, and this one is pectin. Now, I'm honestly not sure if this is in the book or not, but I did memorize it. I actually googled it and I memorized it. So yeah, but either way, the enzyme pectinase can catalyze the hydrolysis of pectin found in fruits, allowing the easy extraction of juice from fruits. Raising the temperature above 40 degrees Celsius decreases because it causes it to get it causes it to become denatured. At high temperatures above 40 degrees, the something which hold the protein together and determine the shape of the enzyme or protein are broken. So the bonds are called cross linkages. Linkages, I think. I could be wrong, but I'm sure I spelled this wrong or something, but the bonds are, I think, called cross linkages or something like that. I'll see when I submit the online quiz, but basically it's just like the bonds are broken, and which causes the shape to break, which leads to the enzyme losing its function. The enzyme loses its function. See, exactly. No, its shape. And the substrate can no longer fit at the active site. Loss of shape results in loss of function of the enzyme. The enzyme is said to be denatured. This is irreversible. So like the alternative, explaining why the increased temperature up to 40 degrees Celsius results in an increase of activity of most enzymes. Okay, a rise in temperature increases the rate of most chemical reactions up to 40 degrees Celsius. A 10-degree rise in temperature doubles the rate of the reaction. This was in the book. Definitely have to memorize it. As temperature increases, the enzyme and substrate gain kinetic energy. This increases the frequency of effective collisions between the enzyme and substrate. There are higher chances of forming enzyme-substrate complexes. More substrate is converted into product per unit time. The blank is the part of the enzyme molecule into which the substrate fits. Active site. Its shape is compatible oh sorry, oh my gosh, compatible, the shape of the blank and blank of the enzyme are complementary, of the substrate and active site of the enzyme are complementary. The substrate binds the enzyme, forming a temporary enzyme substrate complex. Substrate is converted into the product and released. The enzyme is not used up in the reactant and it is ready to bind to another substrate molecule. Enzymes are known as biological catalysts as they're proteins that speed up chemical reactions but remain unchanged at the end of the reaction. Enzymes contain a cleft or depression called the, I think, active site, because it's like the place, like the cleft, I guess, that binds to another. It's the place that binds the other molecule to the actual enzyme itself, the other molecule being the substrate. The combined structure is known as the enzyme substrate complex. The enzyme is said to be blank for the substrate. Said to be, where is it? Specific for the substrate. For an enzyme mediated chemical reaction, the shape and charge of the substrate must be compatible with the active site of the enzyme. Here, the variable being tested. Okay, so let's just read the thing first. Equal amounts of amylase solution and starch solution were added to 4 different test tubes labeled 1, 2, 3, and 4 and incubated at different temperatures as shown below. Tube 1, add ice and water to keep the temperature at about 10 degrees Celsius. Tube 2, mix hot and cold water to prepare warm water and keep the temperature at around 35 degrees Celsius. Tube 3, use cold water to keep the temperature at 25 degrees Celsius. And Tube 4, you add hot water to keep the temperature at around 65 degrees Celsius. Okay, the variable being tested is the effect of temperature on the action of amylase or enzymes. The product formed as a result of the hydrolysis of starch by amylase is maltose, because when you break down starch you get maltose. We can add blank instead of amylase to starch. This will prove that the hydrolysis of starch is due to the presence of amylase. Could add water, because it's really the only logical thing to replace amylase with. The product was first detected in test tube blank, where the temperature is near the optimum temperature for the action of amylase, which is 40 degrees Celsius. So they already told you the optimum temperature, which is 40 degrees Celsius. And here, the one that's closest to 40 degrees Celsius is easily 35 degrees Celsius. Tube 2. But it is good to keep in mind that anyway, 65 degrees Celsius would mean that the enzyme has already been denatured, and the higher the temperature, as long as it's not above 40, the better, because the quicker the reaction. This was last detected in test tube blank, as the amylase has minimal activity at lower temperatures. Test tube 1 had the lowest temperature, which is 10 degrees Celsius, and the product is not detected at all in test tube 4 because the amylase is completely denatured, because it was heated to 65 degrees Celsius. Now to submit and see if we got anything wrong. Okay, a couple wrong. See this? I think it's specific. So like I said before, same meaning. It's just that the online quiz is very specific. Here. when all the active sites, once again, just the online quiz being specific with their answers here. Protease. Maybe I spelled it wrong or something the first time. Protease. Lipase. I probably just accidentally put something like mistyped or something. The blank is the part of the enzyme. Wait, let me see. Which one's wrong? Its shape is specific. I think so. Specific. Let's see. Okay. So the other ones are correct. I just used the wrong word for it. Here. Okay. Oh, sorry. I was supposed to put it plural. I put it singular. My bad. So it's proteases and lipases instead of protease and lipase. And here, okay, so I apparently spelled complementary wrong. Or no, I put compatible. Okay, so it's complementary. So basically, just watch out for the specific words that you're supposed to use. And other than that, that's it.